Ladies and gentlemen, boys and girls, this year's prize celebrates theoretical chemistry as a tool for understanding, predicting, and indeed designing complex chemical reactions. This is important because understanding is important, and it's useful because design is useful. Now, theoretical chemistry has, of course, always been important and useful, at least to theoretical chemists. But through the work of this year's laureates, Martin Karplus, Arieh Warshel, and Warshel Levitt, theoretical chemistry has become a tool not only for those in the trade, but for all chemists, biologists, medical people, and those involved in material science. It has simply become one more tool in the toolbox of science, and for that, we are very grateful. The first speaker is Professor Martin Karplus. He was born in Vienna in 1930. He received his PhD in 1953 from Caltech. And he is Professor Conventionné at the Université de Strasbourg in France, and also Theodore Williams Richard Professor of Chemistry Emeritus at Harvard University in Cambridge, Massachusetts. Please come onto the stage.
Paraphrasing Ralph Waldo Emerson, I shall try to show in this lecture how I've gone where there was no path and left the trail. Now, in trying to apply computational methods to complex systems, the essential idea has been to introduce classical concepts wherever possible to replace the much more complex quantum mechanics. And if we look at something that Dirac wrote in 1929, which many of you are familiar with, he said, the underlying physical laws necessary for the mathematical theory of a large part of physics and the whole of chemistry are thus completely known, and the difficulty is only that the exact application of these laws leads to equations that are much too complicated to be soluble. Many people know this, but they may not be as familiar that this was only the first part of a paragraph which went on. It therefore becomes desirable that approximate practical methods of applying quantum mechanics should be developed, which can lead to explanation of the main features of complex atomic systems without too much computation. This could be regarded as a leitmotif of this year's prize in chemistry, but actually what Dirac was thinking of was not classical mechanics, but simplifying quantum mechanics. Now, in trying to study more complex systems, we really have two aspects that we have to consider: the potential surface on which the atoms move, and the laws of motion that govern the atoms, and both of these we would like to treat with classical methods where possible. Now, the Nobel Prize focused on the potential surface, and I will, partly to save time, I won't read everything that is said here, But basically, the point is that both Michael Levitt and Arieh Warshel come from the Lifson group, which was one of the pioneers in this area, and they will probably tell you about the potential surface simplifications. What I would like to focus on is the laws of motion for the atoms. And repeating in some sense, although the laws governing the motions of atoms are quantum mechanical, the essential realization that made possible the treatment of the dynamics of complex systems like proteins was that classical mechanical description was adequate in most cases. And from my own perspective, this realization was derived from simulations that we did in the 60s of a very simple reaction, H plus H2 exchanging atoms. And just to make this clearer, we have 3 atoms, a molecule AB and an atom C. The atom C collides with the molecule and leaves a new molecule, BC, and an atom A. Now, in order to do these calculations, we of course needed a potential surface, as I've already mentioned. And what we used here was a semi-empirical valence bond surface, as it's called. I was a student of Linus Pauling, and Linus Pauling believed that valence bond theory could solve everything. And so Dick Porter and I developed what has now become known as the PK surface, which is quite accurate. It's not perfect, as has been shown by later calculations, but I show down below here the energy as a function of the reaction coordinate, and the essential point is that there's a high barrier to go over for the reaction to take place. Now, I've shown the linear collision, which happens to be the lowest energy collision, but of course the actual reaction takes place in 3 dimensions. And let me just show you here, we are using classical mechanics to describe the reaction, and since there are only 3 atoms present, we can describe the reaction in terms of 3 coordinates, the 3 distances between the atoms, which is shown here on the left. And it's the distances between the atoms as a function of time in femtoseconds, which turns out to be the reasonable time scale. And as you see in yellow is illustrated the region where there's strong interactions. This is a reactive collision. And so, as you see, the molecule AB comes in, and the atom C comes in here. And at the end, you have a new molecule formed, the BC molecule, and the atom A goes out. Now, shortly after this, in 1967, with some primitive ray graphics as was available at Harvard, we made this film which shows the reaction. And you see the molecule vibrating, the atom coming in. Whoops, I guess, sorry. Hopefully this will remind you of what I've said already. Here again, you have the atom, the molecule vibrating, the atom coming in. It interacts with one of the atoms and goes out again with vibrational excitation. In the next slide, we have a non-reactive collision, again in 3 dimensions. Again, the interaction time is short on the femtosecond time scale. And you may ask your question, why are reactions even at high temperatures, like 1,000 degrees, which is the reasonable temperature for this reaction, why don't they occur on the femtosecond time scale? And the obvious answer is the reaction rate is determined by a large number of collisions. Most of these collisions don't have enough energy to get over the barrier. And so even though an individual event is very fast, the overall rate constant is many orders of magnitude slower. Let me show you here this non-reactive collision which is, see there's again this interaction region, and the interaction is such that the molecule is highly rotationally excited. Now I want to mention one thing, a problem that arose when we tried to show this. Obviously what we're trying to do here is to make the atoms go larger as they come forward and become smaller as they go backward. And the question was, how do you introduce this perspective? Now this has nothing to do with the Nobel Prize, but it's something that I'm proud of, is to find a good law for showing perspective. And what I did was to visit Venice, which I actually did many times, and looked at the paintings of Canaletto and the actual distances and how he presented perspective. And it turned out that he used an exponential law, e to the alpha r. I don't remember the alpha. And so if I had published this, there are many things that I did which were not published, but maybe there would be a Karplus law in our theory as well as the Karplus equation in nuclear magnetic resonance. Now, one of the things that was essential in my development of these applications was to make sure that this would look like a very reasonable description of the H plus H2 reaction was actually the correct description. Obviously, to do that, you had to use quantum mechanics. And it was about 10 years later that a good friend of mine, Aaron Cooperman's group, and Wyatt's group were able to do an accurate quantum mechanical calculation. Since we had used an approximate potential to describe it, to test the methodology, this was not to test whether it agreed with nature, but to test our methodology, they used exactly the same potential as we had used, and they found that the classical results were as accurate as the quantum mechanical results that they obtained with much, much more work. And so the conclusion from this is that if a light atom like hydrogen atom, where you would expect the largest quantum effects could be described classically in most cases, not in all, there's tunneling and so on, that for the larger atoms, carbon, nitrogen, and oxygen, the heavier atoms, of which biomolecules are composed, classical mechanics should be valid. But before turning to the larger biomolecules, let me go back a little bit to one of the items that was actually mentioned in the Nobel Award. I had been very interested in vision at Harvard, and in vision, the essential element is the photoisomerization of the chromophore retinal, which initially is 11-12 cis, cis meaning that around this double bond, the organization is as indicated, and when it photoisomerizes, it goes to 11-12 trans. And this photoisomerization is, if you like, gives the kick, which starts the visual cycle. Now, one of the things that we wondered about in the 11 cis and the chromophore ready to be photoisomerized, you have the possibility that around the 12-13 bond, it can be cis, it's called S-cis because it's a single bond, or it can be S-trans. And if you just look at the two, you would say, well, you don't need any high-level calculations, that obviously this one is going to be lower energy than this. Now, it actually turns out when you do the calculations, because the single bonds can twist very easily, these are very close in energy. Our calculations showed that this was lowest in energy, and this had various implications for the visual cycle, so we submitted it to Nature, and of course immediately got back the answer, well, there are no experiments that show that it's correct, so how can we publish it? Not realizing, and this is of course still true, that once there is an experimental result, then you're no longer making a prediction. And so I was sufficiently annoyed about that, so I wrote John Maddox, who was the editor of Nature, and said, look, you should really publish this, it's important. And he, after maybe an hour of discussion, said, okay, we'll publish it. We'll see what happens. They published our prediction that this was the lowest energy chromophore. And fortunately for him and for us, about 6 months later, an X-ray structure by Carl and Carl was published, which confirmed our result. But, so let's go on now and talk about the isomerization, which Warshel, when he was still a postdoc with me, we developed the method of using classical trajectories of the type for H plus H2 with a surface crossing model to be able to treat the photoisomerization. Here's a very simple case, 2-butene, where you have the two methyl groups on the same side of the double bond. After the light strikes, you get the trans-butene with the two methyl groups on opposite sides. Now, one thing that Warshel did very shortly after he left was to have the idea that in the protein might restrict the system from isomerizing if the two ends had to move. So he did a calculation which is illustrated here. He basically fixed the two end groups, threw away the rest of the protein, and then said, how can this isomerize? And he came up with this very clever bicycle pedal model, in which case you could get isomerization without moving the end groups. More work has been done on this since then, and it shows that the isomerization is similar to what he suggested, but it's somewhat more complicated. And when you actually include the protein, the rhodopsin, the protein relaxation plays a significant role. At the same time, actually, in 1976, we did the first calculation applying the trajectory methods to a protein. It's a small protein, has 58 amino acids and on the order of 500 atoms. We used the potential, was slightly modified from the Schrager and Lifson group potential that was developed by Bruce Gellin. And we did essentially exactly the same thing as if we had done for the 3-atom H plus H2 system, except we now had 500 atoms. But still, classical equations of motion are applicable, and so it was a very natural generalization. Now, I should mention that other people, I guess, hadn't worried as much as I did about whether the classical mechanics was applicable, but a pioneer in molecular dynamics, Anis Rahman, at about the time of the late 60s, had done already some simulations of liquid argon. And so this simulation of this very small, uninteresting protein, we still don't know exactly what its function is, gave results which the graphics weren't good enough so that we could actually illustrate what happened with the graphic systems. And so Bruce Gellin made this drawing of the inhibitor at the initial stage and after 3.2 picoseconds. And if you look, you can see that the structure is essentially the same, but everywhere the residues, represented by a single point, have moved. Now, to bring us up to date, Viktor Ovchinnikov, one of the members of my group now, took essentially the same system with a slightly different potential, and we see that there are these local fluctuations everywhere which occur in a system like this. And in an oral history that Andy McCammon had recorded somewhat later, he had made this wonderful statement, which is, there was a sense, even at the time, of something truly historic going on, of getting these first glimpses of how an enzyme molecule, for example, might undergo internal motions that allow it to function as a biological catalyst. Obviously, what we know nowadays, where there are thousands of simulations of this type being done, that what we felt at that time was really the beginning of a new era in the understanding of biological systems. Now, of course, as computers improve, one could improve the simulations, not only by improving the potentials, but also by doing longer simulations. And it was in '88, about 12 years later, that Levitt and his student Sharon did a 20 times longer simulation. And what was very important, they did it in a box of water molecules. Obviously, proteins, well, they don't exist really in water. It's a rather complicated cellular fluid. But nevertheless, this is a better representation than the vacuum one that we had to use in the early work. And they, I would say, qualitatively, they confirmed what we had found. They found that the motions of the residues were slightly smaller, and that because of the water friction, they were slightly slower. Then, much more recently, with specially designed computers, Shaw and his co-workers have done a simulation that's 1 millisecond in length. And to us, I mean, not very much interesting happens. You get somewhat more global motions in the protein. But to us, what the most important thing was the following. We always were scared, I suppose is the correct word, that these potentials, which worked for 10 picoseconds or 100 picoseconds, if you simulated the system for a millisecond, it would just fall apart. And the important result, as I say, from my own point of view, was that the protein was perfectly happy, and even with these crude classical potentials, the system worked as it should. I mean, it's a very difficult thing in this area to be able to check that you are correct, because there are no detailed experimental data. I mean, we do need experiments still, in spite of what the Nobel citation says, particularly to be sure that what we're doing is meaningful. I won't have time to talk about protein folding, which is another very interesting area nowadays. But then we're going to go on to the next slide, which reminds me that, these motions are present in proteins at room temperature. And like everything else in nature, nature will have made use of them by evolution. And there's this very well-known statement: everything that living things do can be understood in terms of the jigglings and wigglings of atoms. But you might be interested that 2,000 years before, a Greek poet, Titus Lucretius, made the following statement: the atoms are internal and always moving. Everything comes into existence simply because of the random movement of atoms, which, given enough time, will form and reform, constantly experimenting with different configurations of matter, from which we will eventually merge everything we know. Now, what exactly he meant by that, I have no idea, but it really is an amazing statement. And he never wrote anything else, but there it is. So the final part of the talk, really, I would like to indicate how nature has developed the structure of proteins to be able to use the motions to put them to work. And in a qualitative sense, there's sort of two parts to this. I mean, evolution determines the protein structure, and it's a structure in many cases, not in all cases, where the protein is made up of partly rigid units and then hinges which allow the rigid units to move one relative to the other. and then there's some sort of signal, usually the binding of a ligand, which changes the equilibrium between one structure and another. And let me show you a particular enzyme which has been studied by many people, but what this enzyme does. Here you have adenosine diphosphate. Two of these come together in the enzyme, and one of the phosphates is transferred to the other one, so you have adenosine triphosphate and monophosphate. And like many enzymes, this one has an open structure which permits the substrate to come in and, of course, the product to go out, and a closed structure where you have a sort of reaction chamber which is isolated from the surroundings where the reaction can take place. And this is, again, a cartoon of what happens. As the two substrates come in, you have the open structure. It closes onto the substrates, and then over here is shown that the reaction takes place and the products go out. And it's an interesting point in this reaction and many others. The chemistry has been optimized so much that the chemistry is no longer the rate-determining step. These are perfect enzymes in what Jeremy Knowles' way of describing things, that the rate-determining step of the overall reaction is not the chemistry, but rather the opening of the system to let the products escape. Now let me finally turn to one particular system that's been of great interest to us. Molecular motors are, of course, the sine qua non example of how the jigglings and wigglings are put to work to do something that nature wants and needs. And we've studied several different motors, but I will talk just about one of them because, of course, the Physiology and Medicine Prize was given about the function of vesicles, which provide nutrients and other materials which are necessary for the function of the cell. Now, one point that wasn't stressed in these lectures, not all vesicles need to be moved from one place to another. But kinesin, which was discovered in 1985, is very important in the sense that it's a molecule that does, in fact, do the transport of vesicles. The vesicles are up here. We know almost nothing about their structure or how they are attached. But it's a molecule, well, it's actually two molecules that have each one has this globular foot, as you will see, and then they're tied together by a strand to form a coiled coil which holds the vesicle. And what the Physiology and Medicine lectures were mainly about is how the vesicles open and close to give up their cargo at just the right time. This is concerned with how they move around. And again, this is a cartoon, and we'll just watch it. The microtubules down below, and they're the two globular feet. And here's a real-time photograph, enlarged about 100,000 times, where the kinesin molecules are actually moving on the microtubule. But I now want to focus more specifically on how kinesin functions. And if you look carefully at this, you can see that kinesin walks in the sense that we walk. It puts the left foot forward, and then it puts the right foot forward, and then the left foot forward. Now, so that it is walking in one sense, but of course, it's not surprising that these two molecules tied together don't walk really very well. And to me, what the way they're walking is much more like a person who has artificial legs, who sort of walks forward like this, but that a molecular system without all the things we have to make our feet work can do this is amazing to me. Now we'll look at a little more detail, and without going into all of it, we can see, here we'll follow this next power stroke. All the products of the reaction are important, but ATP coming in, which you just saw coming in, is the key to throwing forward the back foot to take another step. And here is now an X-ray structure of the molecule, which again has obviously the two globular feet connected by this beta strand to form a coiled coil here. And we did calculations which showed that just having this one beta strand would not be a system sufficiently rigid to be able to do this power stroke. And so the question was how it happened, and then we realized that there was another beta strand here, the N-terminal one, the initial one, which was disorganized in certain structures but could form a two-stranded beta sheet. We call this the cover strand, and the two-stranded beta sheet the cover neck bundle. And in simulations that we did, which are shown here, the work of Wang and others, we see the two globular feet and the disordered cover strand, all of this one sitting on the model of the microtubule. The ATP binds, and the simulations show that you form this two-stranded bundle, which looks very much like a spring. If you look at the details, it's a high-energy construct, which the simulations suggest, in fact, acts like a string and throws forward the back foot to give you the beginning of the next step. Mutation experiments have been done to support this model, but we're still working on trying to show that it is really correct. So just make a few remarks about the Kinesin motors. They're very important. I mean, if they don't work, mitosis is inhibited because kinesins carry things very backwards and forwards. Obviously, axonal transport, and kinesin was first discovered in the giant axon of the squid. So the vesicles can't get to where they're supposed to. And finally, it's sort of interest that viruses, who also are very clever, have learned that if they attach them where the vesicles are, the kinesin following the microtubule roads in the cell will get them across the cell in a few minutes instead of the 10 hours that it would take by diffusion in the complex cellular medium. So what does the future hold? Obviously, we all know that predictions are hard, but these are pretty conservative ones. And one thing that was already mentioned in the introduction and was really sort of my dream, if you like, when we started doing these simulations, is that not that they would take over from experiments, but rather that experimentalists would use them as a tool, just like X-ray or NNR or whatever, to study systems and get a better understanding than they could with either one type, experimental type or theoretical alone. And that is certainly beginning now, and the hope is that the Nobel Prize, which says what we've done is something important, will aid in this. In terms of actual simulations, people are going toward more complicated systems, simulating viruses, ribosomes, cells. If I were 30 years younger, I would be simulating the brain, but I'm not. And so this future, we hope we'll see. Some of it is already in progress. But I, again, want to caution, as I do my students always, that what they're doing has limitations, just like do experiments, and they have to be careful, particularly when they find something interesting, that there isn't some error in what they have done. Finally, let me show you the people to whom this lecture is dedicated. These are the Karplusians, 244 people who have worked in my laboratory, if I can call it that, in Illinois, Columbia, Harvard, Paris, and Strasbourg. And it's to them that I dedicate this lecture, because without them, I wouldn't be here. And finally, let me mention one other person, my wife, Marcy, who is down here, who was willing to live with a person who spent all his time working, but even more than that, she was brave enough to be my laboratory administrator and made possible our movement from one part of the world to another. And in preparing for coming here, the complexity of everything that one has to do to be in the right place at the right time, I would have never kept up with it if she hadn't been there. Thank you very much.
Well, thank you very much.
Thank you.
Brilliant lecture. Our next Nobel laureate in chemistry, 2013, is Professor Arieh Warshel. Professor Arieh Warshel was born 1940 in the kibbutz de Nahum in Israel. He received his PhD in 1969 from the Weizmann Institute of Science in Rehovot in Israel, and he is distinguished professor at the University of Southern California in Los Angeles.
Hi. Okay, so I would like to thank the Nobel Committee and the Royal Swedish Academy for bestowing this great honor on my colleague and me. And I will try to describe in this talk, there will be 3 short parts. One, the overall motivation, a short historical perspective, and then demonstration on enzymes and on more complex systems. So don't get scared. In our body, there are cells. You have hormones and other things that tell the cell what to do. And my main point is that all of these junctions, or most of them, are proteins that you would like to understand, or to try to understand, how they work. And the good news is that since the late 50s, after Perutz and Kendrew solved the first 2 proteins, there was huge advance. There are a lot of structural information. From my perspective, very important were the solutions of structure of enzymes, lysozyme by D.C. Phillips, and chymotrypsin by David Blow. And one of the key questions is, how could you use the structure of enzymes to understand how it works? Essentially, the center of at least my interest is how different complex molecules are working. So we have the structure, and for the people who don't know what are enzymes, okay, on my screen, you did not see it, but it chops molecules much, much faster than the same reaction in solution. And the question has been for a very long time, what is about this biological construct that makes it work so fast? And at some stage, at least I concluded that it is too complicated, despite the enormous information, knowing the structure and knowing how fast it works is not enough. And one way to realize it is that if you have a clock, you could see the clock, you could see how fast it works, and some of us, I mean, there are people like Ehud Barak in Israel who knew to assemble clocks, but most of us don't know how it works. And I would liken the biochemistry as kind of finding the clock. Crystallography shows all the parts. A single molecule spectroscopy shows us how fast they rotate, and you still want to understand. So my introduction to this problem was relatively early. During my undergraduate work at the Technion, I had a project where I tried to figure out the speed of chymotrypsin by NMR, which essentially I succeeded, but we submitted an abstract. I did not want it to be published. And the conclusion was that enzymes do not work by electrostatic effects, which, of course, is extremely incorrect. But this was the conclusion. You change the salt concentration in solution, nothing happened. Therefore, electrostatic is unlikely to be important. I had another glimpse into enzymes. I took physics course with the physicist from very complicated book about collisions. I did not understand most of it, but what I got from the class was that there are always solutions, most of them due to Max Born, which are asymptotic solution. If you know what happened at the beginning, at the end, you have a good chance to figure out what happened at the complex part. And I told a classmate of mine that one day I will develop a asymptotic wave function for enzyme. I did not know how. And when I finish the Technion, I have to choose where to go. And in contrast to Mike Levitt, who saw TV programs that choose science, I never really saw too much what to do next. But I read a news article about Schneur Lifson, who became the scientific director of the Weizmann Institute. And my only information going to his group, which was only me, was that his kibbutz near David was 3 kilometers from my kibbutz. And I thought it would be interesting. So by the way, this is again a true story. So I joined him, and during like 66 to the end of 69, we were involved with developing what became called the consistent force field, and with the great help of Mike, designing a program that allowed to do molecular mechanics, which means to treat large molecules as ball and springs, and to get the actual solutions. So this molecule could describe vibrations, structures of crystals, and so on. And Mike used it later to minimize proteins. So this was the first stage, seeing that you could model almost anything by ball and springs. Near the end of this work, I also experimented a little with valence bond, but when I moved with Martin at 1970-72, we developed this QM plus MM for retinal and other conjugated molecules, where you model the pi electrons quantum mechanically and the sigma classically. So it's combining classical force field with quantum descriptions of delocalized electron. It's still quite powerful approach. And then when I returned to the Weizmann Institute, I decided to go back to try to do enzymes. I wrote a program that nobody except me ever used it, which described all the atoms quantum mechanically, all the electrons, and then combine it with molecular mechanics program that Mike was using to model lysozyme. And I basically thought that I will just put the substrate of lysozyme, which is a sugar molecule, inside the enzyme and the bond will break. And after a few months, I found that my enzyme is much slower than any real enzyme. Essentially, no bond is ever being broken. And the story was, after a few more months, that the major effect of the enzyme was missing in this combination of quantum plus molecular mechanics. It appears that when you break chemical bonds in solution, it usually breaks to plus and minus, and you need the environment to tell the quantum that it is good to be plus and minus. All of this is one line in the code, but it was not done before. And with this, there was the emergence of the coupled QMMM, which allow you to look at a protein. So in principle, you have small part by some quantum mechanical programs, the rest by molecular mechanics, and you hope that it will let you model proteins. This is part of the overall approach, I don't know if it's working, of focusing on the important part and having the rest with less pixels. And eventually, I found out that it will take 30 or 40 years until the original QMMM will give reliable result with free energy. We could do it only now. And I moved to a valence bond descriptions similar to what Martin described, but with the environment, the solvent, influencing the initial and final state. Now, this is exactly the asymptotic wave function I was talking about from 65. And this appeared to be extremely powerful. And the main reason why it was so powerful is that you train the enzymatic reaction by studying chemistry in water. So we did not have to invent quantum mechanics for the reaction. We calibrated on chemistry in water, and then we removed the water and put in the actual protein. I'll just show you very few examples. This is a protein that involves in signal transduction. When it is defected, you get cancer. So this is a simulation of a reaction. There is a water molecule attacking phosphate and breaking phosphate-oxygen bond. Nothing happens as usual. Does it move? Okay, it should move by itself. Anyhow, it eventually breaks. But the point is not to produce such movies, but to actually calculate the energetics of the reaction in different systems. And it works quite accurately. You could predict the effect of mutation. In later years, we specialize more in mutating the protein. This is from Jen Wang, who was both very, or is very good artist and very good scientist. So you basically mutate the protein, and it does give you quantitative results in many, many cases. So you could predict what will happen with mutation that has general applications. We also use this approach to actually solve the secret of enzyme catalysis. It's harder to understand than what most thinks, because actually the enzyme work so fast, not by interacting very strongly with the broken bond, but by not spending energy changing its structure when you move from the reactant to product. So this is called pre-organization effect, and I encourage you to read. We don't have time to discuss it. So now we move to the second part. So we know to do enzymes, we do it, we try to do enzyme design. And now there is other part, how do you treat large systems and long reactions? Now when you have very fast reactions, it's really easy to use atomistic molecular dynamics and to see what happens. In one example, you solve Newton's equation of motions, you saw Martin early trajectories. When you apply it to the visual pigment, you produce a movie. This is the 76 work, which was done in basically Walt Disney animation. And what happened is that after absorption of light, this one refused to move. Okay, you will see in a minute. So this is the ground state. Light will come eventually. I know it because I saw the movie. The light will come from here. And the molecule will rotate from cis to trans. And it happens really, really fast when the light comes. It rotates without changing so much the structure, so it's a very restricted motions. Eventually, when it's finished to rotate, it pushes the protein. Protein change structure, activate G protein through, it's like the GPCR, which is last year's Nobel Prize, and it's eventually visual signal. We use similar approach for photosynthesis, and now we try to ask what happens when the time is long. You have to do something. Of course, computers become much stronger, but at present there is a lot to be done with simpler models. So the trick is to use what are called coarse grain modeling to simplify the atoms. The beginning of this was work with Michael, where we replace the atoms by the side chains by spheres. In more recent years, we felt, learning from enzymes, that electrostatic is the key to structure-function correlation. We trained this initial 75 model to give us reliable energies. And it took telling charges how much it costs to move from water to the protein. It took telling charges how much they like each other. After doing it, and you will see how basically nice energetics it produces, we also have the questions of how do we model reliably motions in very long time, milliseconds, even seconds. So this is regular all-atom molecular dynamics. This is supposed to be fish. Let's do it again. Yes, so when you try to simulate fish, you don't simulate everything around it. You simulate it as effective random motion or as a friction. So the trick is to find the correct friction for motion in a protein. And there are Einstein theory, Chandrasekhar, all of them. None of them actually give you the correct friction because there is always issue of memory. So we invent approach which we stole the name renormalization for it. And the idea is as follows. You take a full model. This is the adenylate kinase you heard in the previous lecture. But we don't want to wait until it will close. So we apply to it very, very strong force and force it to close relatively fast. Then we take a simple model with friction. We apply the same force and adjust the friction until both the full model and the simple model move at the same speed. Once this is done, we remove the force and just let Brownian dynamics work on the simple model, where we tend to believe the results. So we could produce, in principle at least, single molecule and NMR results, and we could ask questions. And I will show you now in the next 4 examples what we could do with such a model. So there is a ATPase. It's kind of the hydrogen atom of motors. And one of the nice questions about this guy is that when the central stalk rotates, it turns these units and force the reactions of ATP to ADP to occur. Now, here we have plenty of structural information, not of all the intermediates, but basically more than any other system. Beautiful single molecule experiment of Kinoshita and others, which have very interesting conclusion. Instead of rotating 120 degrees in each part of the cycle, it rotates 80 degrees and rest, and then the chemistry occurs. So the question is, could you use the structure to figure out why it happened? We use this electrostatically enhanced coarse-grained model, and already in the first attempts, first we got this nice direction, the energies are in the right range, and my postdoc, Cheyenne, found that at 80 degrees, because of electrostatic effect, it does not want to rotate anymore. And we end up with what looked like a complicated diagram. I remember that Mike once asked me, could you tell me in 1 minute how ATPase work? This is the answer, but it's more than 1 minute. What happened is that when you try to hydrolyze ATP to ADP in water, it would take you months. The barrier is very high, and there is nothing useful to do with it. When you put it in the protein, it rotates up to 80 degrees. Then the barrier is reduced drastically, and in milliseconds it rotates. Then the phosphate goes out. You go downhill in energy. This is the energy of life. This 10 kilocalories is what drive living processes. And this type of model allows you to ask many questions. We also ask other questions on proton transfer in the lower part, but we won't talk about it. Now, you heard about this type of, in this case, it's myosin 5. It's walking in one direction. And because our coarse-grained model is powerful in producing energetics, we try to ask what to me is the key question. Why it goes in one direction? I know that this one, I will have to go out of this just a second. Okay, so usually when you walk similar people or the same person forward and backward, they walk in the same speed. So you, I mean, no problem. So this does not happen in daily life. And the question is, why it happened to myosin 5 and essentially to all of these systems? What forced them to go in one direction? And there are structural models. We put them in our model, and we found that we get electrostatic energy of 11 kcal per mole, which, by the way, agree with force experiment, that when the knee is bent, it's easier than when the knee is straight. And as much as we are concerned, in all this complicated cycle, which become much more complicated here, and in the paper it's even more complicated, when you go through it, you find that going backward it's much harder to go backward than forward, and you could ask many questions along this line. So this is an example of using a simplified coarse-grained model, focusing on electrostatic. We could also run Langevin dynamics and ask other questions. And now, how much time I have? Where is it? Okay. So now I will just give you two more applications. We could use the same philosophy to ask many questions. There are voltage-activated ion channels. This is Rod McKinnon, who got the Nobel Prize for it. And there is this fascinating issue that when the voltage is like this, they are close. When you change the voltage, maybe let's see it again. It become open. And the question is, could you simulate it? Now, there are simulations with this DE show computer, but it's hard to know what really happened. And the problem is, at least from where I'm coming from, I do not believe continuum electrostatic, so I have to see everything. And we felt that we must model the electrodes, the electrolyte, and all of this, so we done it. And this is an older movie of without the voltage. These guys are the potassium. They are in different colors, so you could follow them. And these are the chlorine, so we could model this. And the more exciting thing is that now we could understand the early current in such systems by doing what I think are sufficient simplifications, still keeping the physics. The last things for 1 second, there is the ribosome, which is very complicated by itself, but to make life much more complicated, Gunnar von Eine, who is here, connected a faulty peptide, which usually stuck in the ribosome, to the translocon, and sometimes the translocon helped this guy to be pulled in. And to model this, you have to, people talk on multiscale, but you also need multi-talent. Not me, but you have to know the art of doing many things. So here you have to do the chemistry to see why it's stuck, to model pulling and all of this, and it ends up with a nice movie of how a faulty nascent peptide tried to move through the ribosome, move to the translocon, and then, okay, eventually, there is very new paper in Nature, essentially, that confirm our idea that here it folds before it goes to the translocon. Okay, I don't know how long we could wait, so let's leave it. And the last things, if you ask on the future, one of the things which at least interests me is the issue of drug resistance, trying to predict what will be the next move of a virus or another pathogen. And just very shortly, the idea is that we could combine our ability to look on drugs which the virus try to evade by mutating, and to add to it the fact that the virus does not want to change groups. This is the enzyme of the virus, it's not the virus. The enzyme of the virus still want to do their work, so you cannot take this group and change it. It will help not to bind the drug, but the virus will die because it cannot do its work. So this restricts drastically the possible mutations, and you could try to see what will be the next mutation. Okay, so basically, to summarize, my view of structure-function correlation is that it's mostly electrostatic. So we show you again the bunny. And you have to focus on this. And these are, I cannot acknowledge all, these are people who done large part of the work, and I thank all of them, and I was told that, of course, that I have to thank my wife. No, she deserves it. And thank you very much. Yes.
Thank you very much. Don't fall off the stage. And then I would like to welcome onto the stage the third Nobel laureate in chemistry 2013, Professor Warshel Levitt, who was born in 1947 in Pretoria in South Africa. He got his PhD from the University of Cambridge in 1971. And he is now a Robert and Vivian Kale Professor in Cancer Research, Stanford University School of Medicine in Stanford in the USA. And he picked up citizenships in all these places, I understand. So, welcome onto the stage.
Thank you so much to the Royal Swedish Academy of Sciences and the Nobel Foundation for electing me. It's a great honor and privilege. Thanks too to the speakers who went before me. We didn't actually coordinate our lectures at all, but I think it actually worked out really well. And some things have been said before, other things haven't, but we have very different styles, which is fun. So I am going to speak about the birth and future of multiscale modeling. I think it's also interesting that Martin had a green laser, Arieh had a red laser, and I have a blue laser. So my talk is going to deal with 4 different things: how it began, the birth of computational biology, some new work, future work on multiscale modeling, and then just finally some general things. So how it began. I think it's really important, if you want to get ahead, is to stand on the shoulders of giants. This is always good advice, and I think I was very lucky. Linus Pauling was an enormous influence. I actually gave the Pauling lecture at Stanford a few times and met Linus in his 90s. Another huge influence on me was Francis Crick. He was one of my mentors at Cambridge when I went there in 1968. He was the person who solved the structure of DNA, but really also combined modeling and experiment, and should have given, together with Pauling, respectability to modeling. It didn't quite happen so easily. Then we have experimentalist John Kendrew, played an enormously important part in my life. I'll say more about that. It's also interesting that in the 60s, the most important journals were not Science and Nature, they were Scientific American, because it was the only journal that allowed you to publish colored pictures. And John Kendrew had a cover picture in Scientific American in 1961, and here is Max Perutz, who was in fact John Kendrew's PhD supervisor. Max wanted to solve the crystallographic phase problem with heavy atoms, and he did. For me, Max is the real hero of structural biology, a really amazing person. Finally, David Phillips, the only one of this group who doesn't have a Nobel Prize. I guess Pauling has two, so it sort of compensates. David Phillips was again has a cover picture in Scientific American, a remarkable man and a really early supporter of computational biology. In the Scientific American article, he shows proteins folding up, done by cartoons. This was a very, very early influence. Okay, another really important influence in the field was Los Alamos and the Manhattan Project. The Monte Carlo method was actually invented there. And here I think is a rather kindly picture of Edward Teller, the father of the H-bomb, and I guess the third author in the Metropolis Monte Carlo method. And finally, and Martin did mention Anansi Raman, who actually passed away relatively young. He had done simulations of argon, and then together with Frank Stillinger, simulations of water. And in many ways, because proteins have many atoms, they are much more like liquids. At least they were more like liquids early on than other things. Okay. So I've told you a little bit of how it all began. So this is basically the scene as it was in the mid-late 60s. So now a little bit about the birth of computational structural biology, which is sort of the field that was recognized and is now an okay field to talk about. So John Kendrew, who had won the Nobel Prize in 1962, had an enormous influence over me. He made a television program in 1964. I had come from South Africa at the end of 1963. I was 16 years old, and I'd never seen television. In South Africa at that time, there was no television. And my uncle and aunt with whom I stayed had a early, I call it black, not black and white, but black and yellow television. And on that, I actually watched two things that I remember very clearly. One was the Winter Olympics of 1964, and the other was John Kendrew's program called The Thread of Life, an introduction to molecular biology. And if one just looks at the titles of the various topics he talked about, it could be a current course in molecular biology. I tried to get a copy of the lectures, and I don't think they exist anymore, which is really a pity, because this was one of the first lectures, but it was very influential on me. John Kendrew then actually sent me to Israel to work with Arieh and Shneur Lifson. And the reason he sent me to Israel was I wanted to start a PhD in Cambridge, and he said there was no space, and after some negotiation, he said, okay, if you go to Israel for a year, then we'll take you. So he sent me to Israel in 1967 to work with this man, Shneur Lifson. This is how Shneur looked back in 67 and sort of how I looked at about that time. This is actually a bit later. I looked significantly younger at that time. Shneur really was the person who got all of this started. He was, if you were to delete one person, if you deleted Shneur, my guess is this field would have been delayed very substantially. And he did this essentially by having a philosophical idea. So again, just to recap, at this time, the mid-60s, we knew that biology is all detailed interactions. Arieh showed a clock as the insides. For me, what was remarkable in seeing these papers in Scientific American was that the insides of cells were not like a bunch of blood or soup. They were actually like clockwork. Molecules have shapes, and they function because these shapes move. And that's exactly like a watch or a motor car, or almost anything around us. So in some senses, a really good model for a cell would be a city. New York is a great model for a cell. It's not, a bucket of blood. And this just shows the myoglobin structure, where this heme group binds oxygen, the lysozyme structure that was very influential afterwards, where there's a hexasaccharide here and is bound and cleaved. Okay. So basically, it all started with this philosophical idea of the consistent force field, and the idea that Lifson had was that a carbon is a carbon is a carbon. And this might seem completely trivial and obvious, But at that time, people who were doing analysis of small molecule spectroscopy said that the carbon in carbon dioxide was different from the carbon in methane, and so on. There were many kinds of carbons. Lifson said that the force field, or the potential energy surface for a molecule, needed to be consistent. And the good thing about consistency meant that there wouldn't be too many parameters. So in this paper, Arieh and Schnell basically started to parameterize the force field essentially for small molecules. The force field sort of looks like this. You will notice that I actually have a font which is my handwriting. When I started using PowerPoint, I didn't have that, and I would actually draw things on paper and scan them in. So this is actually my handwriting. So this just shows what the force field looks like. This force field is really interesting because the key thing was that there are many parameters. So for example, if you look at a bond length, if you take a bond and you stretch it, the energy goes up, and you compress it, the energy goes up, just like a spring. The same thing is true of bond angles, torsion angles. They all have these very simple mathematical functions, and in these functions there are two kinds of things. So for example, if we take, say, the bond length, we have the bond length B. The bond length B depends on where the atoms are. Then there are parameters, the bond length B0 and the force constant. The bond length is the sort of preferred length of the bond that it vibrates about. Where do these parameters come from? And again, Lifson's philosophical concept was that there would be a small set of such parameters that could be derived in a consistent way. This ended up being very important because one could then very easily take what had been derived for small molecules and apply it to any molecule, with obviously realizing that there's danger in doing that, but the calculations could be done. I often call this the dead physicist's energy function. In fact, I did have dates here, but Hooke, Fourier, van der Waals, and Coulomb all lived at least 200 years ago, and essentially were responsible for each of these terms. What for me is amazing, and I guess Martin did say in his talk how it's remarkable that things as simple as they were are still being used today, but in many ways this energy function is still being used, say, by the D.E. Shaw calculations that are 10 million times or 100 million times faster. Okay, so Martin also mentioned the importance of the energy surface, or the potential energy function. This is just an example to show how all the methods, whether they're energy minimization, shown here is just dropping down to the nearest minimum, here shown in 2 dimensions, or normal mode dynamics, which I guess goes back to Galileo, where you vibrate about a minimum, and again, you can do that in 2 dimensions. Or molecular dynamics, which was used and certainly pioneered on proteins by Mechano-Chemical and Karplus, basically involves moving downhill. I'm going to see that now. Again, in 2 dimensions. So you sort of move along, but you have kinetic energy so you can go over barriers. And finally, the most interesting method, because it came along so late, is the Monte Carlo method. And that basically came with Los Alamos. And again, there should be dates here. I guess I messed up my font, and those dates are not there. But this one, Metropolis, is it was published in 55. It was probably done in like 44. But, Euclid is 325 BC, I believe, and the others are in between. Okay. So the physics behind this is kind of old. The difficult thing was to get it all to work. Now, just to say now about some of the real stuff going on, we felt that the key idea here in the multiscale modeling really goes back to a quote that is actually attributed to Einstein: everything should be made as simple as you can, but no simpler. It actually turns out that there's no real evidence that Einstein actually said it. And if you go to a website called Wiki, Investigator, it actually says that a newspaper person, Roger Sessions, actually invented this quote and sort of attributed it to Einstein, so he would get extra credit for that. So I guess I'm happy to stick with this as being an Einstein quote. But it's also very important because the reason you need to do this is in order to get a calculation to work. It has to be simple enough to do the calculation, but it shouldn't be so simple that it's worthless. And I often use the example of simulation here as the weather. If you had a really detailed model of, say, Sweden, you could probably calculate the weather tomorrow in 10 years' time. But that wouldn't really be very useful. In order to calculate the model the weather tomorrow in 2 hours, you need to simplify your model enormously. It actually doesn't really work very well for Sweden. I've noticed that the weather forecasts for Sweden are really, really bad. They're really good in California. In California, they go onto the radio and they say, the weather tomorrow will be like today, and so on for the foreseeable future. Okay, so very much what the work we did was about simplifying representations, minimization with all the non-hydrogen atoms in 1969, atom groups in 75, all atoms and electrons in 76, and then all atoms and water, as Martin mentioned, in 1988. So in many ways, my work has been about multi-scale work. The paper that Arieh mentioned, again with Schnell, was my first paper ever published, involved an energy function that was applied to proteins. It's interesting how I actually got the coordinates of the proteins. In those days, there was no protein data bank, but David Phillips had sent Natan Sharon, a biochemist in Israel, the coordinates of lysozyme and myoglobin. And the idea was that someone would actually build a physical model. And I volunteered to build a physical model together with Yuval Eshdat, and actually got my hands on the coordinates and then had them typed up and put into the computer. So there was sort of an interesting connection there. The next paper involved coarse-grained models. And this one was with Arieh. And basically, we were folding a protein in a very simplified way. We could make the protein fold up. Now, it became more compact. It didn't necessarily become the right answer. And we did this by combining minimization with normal mode thermalization, which actually meant that you got into a minimum, and then you made the thing vibrate around the minimum analytically and jump out of the minimum. So this was an approach to doing that. Okay. So then Arieh, working with me, made this great move into enzyme catalysis, and it's a cool picture of Arieh. I had to show that, and again, now you're considering the atoms and the electrons. This is the first movie of protein dynamics that was actually made for me by Richard Feldman at the NIH at an incredible expense. Each frame of the movie required 2 hours of the biggest computer that existed in the USA at that time. And remember, you're seeing 24 frames a second. This movie was actually also very highly publicized by Richard. It's BPTI, the molecule that McCammon, Gellin, and Karplus did the first calculations on. And they're actually here 4 water molecules. And the movie is sort of fun because one of the water molecules starts to move out into space. And it sort of looks like it moves around a bit like a little spaceship moving around here. In previous talks, I've made a reference to David Bowie's Major Tom, but most people don't get that anymore, I guess, except for the viral YouTube movie made in space by the Canadian astronaut. Okay, so moving ahead, it was obviously important to do protein dynamics in water. And this was actually very difficult to do. It basically took us 11 years after the first calculations by the Karplus group, because people had done simulations of water. Together with Ruth Shawana in the audience, we worked very, very hard to try to match the energy functions used for the protein to those used for water. One had to conserve energy, one had to truncate interactions smoothly, but eventually we got it, and here's a picture of Ruth. And the important thing was, as Martin has said, that the movement, the protein moves in water, but basically the movement in water, not only was it slower, but the deviation from the starting structure was a lot smaller. Here it's 1 angstrom, a lot smaller when done in water than when done in vacuum. And these letters just show other simulations done in vacuum, all showing quite large deviations. Okay. A little bit later, together with Valerie Daggett, we actually did unfolding of the alpha helix. And this movie, shown in the next slide, was interesting because I actually made this movie to show to Linus Pauling when I gave the Pauling lecture in 1990. And all the alpha helices have the wrong hand because I made the film backwards. In other words, the calculation was right, but the negatives had been filmed and turned over. Actually, this happened at the Nobel Foundation when we were introduced just the other day, so it's very easy to make. And this just shows an alpha helix in water. The water is there for the calculation, but it makes it really hard to see what's going on. There are hydrogen bonds making and breaking, and so on. We're now going to see the structure at high temperature. So this is at high temperature, 473 Kelvin, which is 100 degrees centigrade, and the molecule very quickly unfolds, as it should. Just the same thing again. I think the final sequence is just showing this as a space-filling model, which sort of looks a bit like a python moving around. Remember, there is water everywhere. We're just taking it out for the movie. This is an old movie made in 1990. This just shows the same thing in space-filling. It's actually fun to write the program to do this, because computers weren't that fast back in 1990. Okay. So that sort of ends the birth period up to about, say, well, probably there's several periods, maybe up to 1988. I now want to say something about the future, because I think that getting a Nobel Prize has a real drawback. So it kind of makes you feel this is the best thing you're ever going to do in your life. And that's a really depressing thought. I mean, that's a great thought to have 5 minutes before you die. And maybe I'll fall off the stage or something. But, you know, it's not a good way to live. You need to have things you still want to do. Okay, so I'm going to talk here about multiscale dynamics. And start off by saying that one of the key things about multiscale, you can have simpler energy functions, but you can also simplify a system by having fewer degrees of freedom. And if you think about how you'd characterize a person walking, you can probably have a knee angle, bend, and so on, but you don't have to think about all the atoms moving inside. They're all moving there, but if you said about walking, you'd say there's a knee and it bends and the other leg moves forward, etc. So choosing degrees of freedom is really important. And I'm going to start off by describing several sort of recent calculations of work that is in press or about to be published. This is a study of RNA polymerase, the structure that Roger Kornberg got the Nobel Prize for in 2006. Here we want to try to see the molecule working. This is a large structure, and I'm showing this because Daniel Da Silva, the PhD student working with Shui Huang, who's an ex-postdoc of mine, made an amazing movie, which I'm going to show in the next slide. So this is a big system. It's 500,000 atoms, including the water. And this is just a great movie. Now, this is not made by a dynamic simulation. The clock, even David Shaw can't make the clock move so fast for a system of this size. It's made by what they call Markov state simulation, where you think about many different states and you jump between the states. And we're going to see the molecule. This is RNA polymerase, and what it needs to do is move a nucleotide from here to there. This is a fairly simple thing. It's to do with building DNA on RNA, which is what RNA polymerase does. What I really like about the movie is just, I didn't make it, but I guess like Arieh's movie for Retinal, it sort of has the reaction diagram in the corner. And this is really nice because if you can move the slider backwards and forwards, you can locate exactly when things are going to happen. So now this is going to move across and suddenly move into there. So this kind of shows the reaction, and this is actually fitting quite well what is seen experimentally. I certainly agree with my two previous speakers in that experiment is extremely important. Okay, another thing is involving normal modes. It's actually interesting. I was speaking to one of the winners in physics, and he was saying he was doing normal modes based on group theory. So normal modes are a very interesting idea. They go back, I would say, many, many hundreds of years. And we've just succeeded now in doing normal modes of the entire ribosome with 170,000 atoms. This is a difficult calculation done with Junji Zhang and General Bray, two current postdocs in my lab. They're just about to leave. And this just shows the ribosome. The motion is very exaggerated. These are just the first 6 modes concatenated together, showing them with all the atoms, and exactly the same things calculated with the simple sphere model. And the important thing is that the motion looks very functional. That's all I'm saying at this point. We need to analyze it very carefully. It is very exaggerated, but this is the kind of thing you can do. And this calculation, even with all the atoms, takes 10 hours on my laptop, so it's not a big calculation. It's a way in which you can get the essence of what's going on without doing a very, very long simulation, something that I feel is actually important. The final thing is a Monte Carlo calculation, in this case of RNA, using what we call natural moves. And I'll explain this a little bit in the next slide. This was an idea that Peter Manari who's currently, was in my lab, is currently an assistant professor in Oxford, and Adeline Sim, who is a PhD student and is now a postdoc in Singapore. And they did. And the basic idea is as follows. So here is a bit of RNA. And what we're doing here is we're not moving every atom. We're moving whatever we like. You can go in there and say the movement should be bend this helix back 2 turns, or anything you like. When you do moves like that, you break bonds. You can't help breaking bonds. And the idea is that you quickly fix the errors you make in the bonds before accepting or rejecting the step. And this works extremely quickly. This just shows that you can see, in this case, the movement of a base pair. If you only move base pairs, the whole structure just moves like this. But you can also move entire helices, and then you're able to sample much, much wider changes of motion. So again, this is sampling the space by the Monte Carlo method, the method developed at Los Alamos. And you can basically do any kind of hierarchical mixture of the modes. Monte Carlo is very nice because you don't need any derivatives. The calculations can be done in a very easy way, and computers are fast enough to make this possible. Okay. So I just stuck this slide in today because it's really important to try to give an example of what this is actually useful for. And it's very easy to say, well, one day all drugs will be designed using these techniques, and I actually believe that. But when will one day be? 5 years, 10 years, 100 years? But I do have actually one example, which is antibody humanization. There I am, which was actually something that is an interesting story. So it turns out, just to make this important, antibody humanization are the most important anti-cancer drugs right now. The idea is you take a cancer cell, you inject it into a rabbit or mouse, it makes antibodies against that cancer cell, and then you'd like to inject those antibodies back into a person. Trouble is that these are mouse antibodies, and people don't like mouse antibodies injected into them. So what you need to do is humanize the antibody. Now, this is an old idea. The idea first came from Greg Winter and colleagues in Cambridge, and they basically said that the way you humanize an antibody is you just take the hypervariable loops, the loops that bind the substrate, and graft them from the mouse antibody into the human antibody. When I got to Stanford in '87, it was a small company, Protein Design Lab, and they wanted to actually, I became a consultant with that company, so almost like part of my deal of coming, although I think Roger Kornberg wanted me to come to Stanford so I could be a consultant for the company and not the other way around. And at that time, I had done work in the early '80s on how you calculate, how you build models of antibodies. I know Martin had done some work like this as well. And I had a computer program which, given any sequence of antibodies, would build you a so-so model. Now, using these models, Carrie Queen, who is the main author here, basically looked at the antibody and said, okay, it's not enough to just swap in the loops that are complementary determining regions. You have to also change some of the amino acids behind that. Now, this might seem like a very obvious thing, but when you apply things, it's really important to stick with the obvious. So what they did is they got this idea, they found out, had rules for which residues you had to change, and they patented it. And it turned out that the Winter work, which had come before, had also been patented, but their patent didn't work so well, because the Winter patent said, just change the hypervariable loops, whereas in reality you had to say, just change the hypervariable loops and 3 more. And that 3 more ended up being worth a great deal of money. Currently, the antibody therapeutics business is at least $20 billion a year. And Protein Design Lab actually is getting something like $500 million in royalties this year. That's the good news. There are 2 bits of bad news. I have no part of the action. The other bit of bad news is this is a patent that was granted in 1989. That means it expires next year. So, but still, I mean, I think this has a number of things. It shows how basic research can be turned into applied research if you wait 30 years. And it's a very clear example. It's also clear to say that by the time I was asked to help do this, it was boring. We had a program that could do it. I said, run the program yourself. But it was still very important. It was also very important to see how writing the patent clearly was very important. Okay. So one could say that I suffer from lack of focus. I'm going to move ahead quickly now. Our work has been hugely pushed ahead by technology. Computers have become 10,000 times faster, 10,000 times cheaper, 10,000 times higher capacity, and 10,000 times smaller. If this was a car, a new Volvo would cost $3, go 1,000,000 kilometers an hour, hold 50,000 people in comfort, and park in a shoebox. So with that kind of tailwind behind you, there's no way that you cannot be succeeding. Family support has been really important. My mother, who's actually 98 and will be coming, yes, often I say 89, but it's 98, and will be coming to the ceremony. She's on her way here. And my wife, Rena, who's in the audience. The old saying, I'm sure you all know the saying, but it's not quite what you think. This was sent by Eric Lindell, who is sitting in the audience, to Rena, and it's a saying that she's been saying almost every single day since then, becoming increasingly surprised. Take chances. It's very important in science to take chances. Here is me backpacking across the Grey Pass in Patagonia last year. I'm actually not doing my email. I'm using it as a GPS to see how much further I have to go. It was really hard. Take chances, but don't be stupid. Again, 2 years ago, I went to Utto, rented a kayak, had never been in a sea kayak before, had looked at YouTube for pictures of how people sea kayak and felt that I knew how to do it, rented a sea kayak here in Utto, kayaked about 30 kilometers up to Orno and back again, found a small island over here that I call Paradise. Very beautiful. This is my island, which is exquisite, my tent on the island where I slept, but also really, really stupid because the water was so cold, and this was before midsummers, and if I'd capsized, I certainly wouldn't be here now. So I think it's really important to give advice to young people, be passionate, be persistent, be original, and also be kind and good. You never know when that's going to be important. I just want to finally thank my towering heroes of science, as I call them, Sneeuw Lifson, who was an incredible inspiration, and I just wish he'd been here, but he would be over 100 years old, and I guess didn't make it. Then my mentors in Cambridge, John Kendrew, my PhD supervisor, Bob Diamond, Francis Crick and Aaron Klug, and Max Perutz, whom again for me is the hero of science. I don't have anything like the groups that either Martin or Arieh had. I would say that the distinctive thing about me was I am not one of Martin's Karplusians. I'm almost the only one who's not. But otherwise, he really did cover the field really, really well. I did have a number of people from lots of different countries, have been well supported. I also want to thank the Nobel Committee in Chemistry. Now, this is something which is obviously super self-serving, but I'm not doing it because it's self-serving. I'm doing it because they worked very hard to carve out a new field, multiscale simulations, and coined this as being a respectable chemistry and biochemical approach. So they have done really, really well. The big winner is our field, but here I'm using the double meaning of the word field in this overlay. This is the American football field at Stanford, where I was asked to appear on the 10-yard line, and 50,000 people are screaming, Nobel Prize, Nobel Prize. Believe me, that is a high which is really hard to equal. And again, my thanks to you all.
So at this stage, I would like to invite all the laureates in chemistry onto the stage. If you will come here, please. Here we go. Just there. Say hello to the audience. Thank you for very informative, but more importantly, very entertaining lectures.